Welcome to the deep dive. Today we are plunging into, well, one of the most fascinating paradoxes in cancer treatment.
It really is a paradox.
Right. We spend so much effort designing treatments to kill cancer cells, but the real magic happens when those dying cells, they don't just go quietly. They need to scream for help.
Exactly. That's the whole concept behind what we call immunogenic cell death, or ICD.
The ideal situation.
That's the goal, yeah. The treatment kills the tumor cell, and all the debris it leaves behind acts like a flare gun, signaling the immune system to come in, clean up, and, most importantly, remember that specific cancer so it can't come back.
And researchers have been mapping all these different ways a cell can die, what they call regulated cell death, to find which one screams the loudest.
And you have apoptosis, which is very neat and tidy, almost polite.
Too quiet. But then you have this other one, ferroptosis. And it's a messy, violent, catastrophic way to die.
It's incredibly violent. The cell essentially rusts itself to death from the inside out because of iron-dependent lipid damage. You'd think that kind of explosion would be the perfect immune alarm bell.
It should be the ultimate activator, but, and this is the core puzzle we're tackling, it turns out that ferroptosis is surprisingly weak at waking up the immune system.
The scream is there, but something is muffling it almost immediately.
So our mission today is to unpack the molecular spy that's sabotaging this process. It's a brand new discovery, a molecule called extracellular GPX4 that works through a totally unexpected receptor, ZP3.
So before we dive in, let's just quickly define the three key players for you. First, ferroptosis. Just think iron-dependent cell death from damaged lipids.
Got it. Rusting to death.
Second, DAMPs. Damage-associated molecular patterns. These are the molecules released by any dying cell that act as a universal danger signal, the flare gun I mentioned.
And the third?
GPX4. This is an enzyme, glutathione peroxidase 4. And what's crucial to know is that its day job is inside the cell, where it acts as a shield to prevent ferroptosis from happening.
So its job is to stop this process.
Exactly. The whole story changes when this internal guardian somehow ends up outside the cell.
Okay, so let's get into the experiment that really exposed this anomaly. The researchers used a gold standard setup, a tumor vaccination model in mice.
With pancreatic cancer cells. They basically took these cancer cells and killed them in a bunch of different ways in a dish.
Some with drugs that cause apoptosis, some necroptosis, and crucially, some with a drug called Aristin to induce ferroptosis.
Right. And then they take that cell debris and inject it into healthy mice, like a vaccine. The idea is, if the vaccine works, it should train the mouse's immune system.
So that 10 days later, when they challenge those mice with live cancer cells,
the immune system should be ready and waiting, and the tumor shouldn't be able to grow.
And the results were, they were incredibly clear.
They were decisive. The mice vaccinated with the ferroptotic cells, the ones killed by Aristin, had the weakest vaccine effect. The most violent cell death created the least protection.
So why? What was going on? The first thing they checked was for those danger signals, the DAMPs.
Yeah, you have to make sure the flare gun actually fired. And it did.
Right.
They looked for a classic DAMP called HMGB1, which is a universal sign that a cell has ruptured.
And it was there.
Oh, yeah. It was released by all the different death types, including ferroptosis, so the initial alarm was definitely sounded. But here's the clever part.
Go on.
When they blocked that HMGB1 signal, the vaccine effect was totally abolished for all groups. That proved HMGB1 was necessary, but it also meant that during ferroptosis, something else must be actively shutting down its message.
Something was working against it, and this is where the GPX4 discovery comes in.
This is the moment. They found that GPX4, that internal enzyme that's supposed to prevent ferroptosis, was being selectively released out of the cell only when it died by ferroptosis.
So it wasn't just some random protein leaking out. It was specific to this one death mechanism.
Highly specific. They tested it across 60 different human cancer cell lines, used different drugs, and the result was always the same. GPX4 was released during ferroptosis, but not apoptosis, not necroptosis, not another one called cuproptosis. It was a unique signature.
Which makes it what they call a ferroptosis-associated immunosuppressive DAMP.
Exactly. It's a danger signal that tells the immune system there's no danger. It's a self-sabotaging mechanism.
Okay, so you have this unique countersignal. But how do you prove it's actually doing the damage? that it's an active brake and not just some inert piece of cellular junk?
Great question. They did two key experiments. First, they used a monoclonal antibody to specifically block that extracellular GPX4.
To catch it before it could deliver its message.
Precisely. And when they did that, the weak vaccine effect of the ferroptotic cells was significantly restored. The immune system woke back up.
Wow. Yeah.
Then they did the opposite. They took pure recombinant GPX4 protein and injected it directly at the vaccine site. And just as predicted, it actively suppressed the immune response.
That's pretty definitive proof. It's the active saboteur.
It is. And they also noticed that another common immunosuppressive molecule, PGE2, was being released by all the dead cells. But the really interesting part
What's that?
was that when they blocked both GPX4 and PGE2, the immune response was even stronger than blocking either one alone.
So there are two independent stand-down orders being given, but the GPX4 one is the one that's unique to ferroptosis.
That's the key therapeutic insight. It's a unique vulnerability.
Okay, let's just pause there for a quick recap. Ferroptosis is supposed to be a loud immunogenic form of cell death, but it's not.
It's not, because it uniquely releases its own antidote, the enzyme GPX4, which acts as a damp to shut the immune system down.
And crucially, if you can block that extracellular GPX4, you can restore the anti-tumor immune response in these models.
You turn the saboteur into a signal.
All right, we've identified the spy, extracellular GPX4. So the next question is, who is it reporting to? Who is the target?
And the target turns out to be the quarterbacks of the immune system. The dendritic cells, or DCs.
These are the cells that are absolutely essential for finding a threat and then going to activate the T cells to fight it.
Right. And they found that when you expose dendritic cells to GPX4, they do something very strange. They stop preparing for battle, and instead they start pumping out an anti-inflammatory cytokine called IL-10.
Which is basically the all-clear, stand-down signal for the immune system.
Exactly. It's a de-escalation signal. And that strongly suggests that GPX4 wasn't just floating around randomly. It had to be binding to a specific receptor on the surface of that DC.
So how on earth do you find one specific receptor on a cell surface?
Well, they used a really powerful technique, an RNA interference screen. You can think of it as turning off genes one by one in the DC to see which one is responsible for receiving the GPX4 signal.
So they're just flipping switches until the light goes out.
That's a great way to put it. And the top hit, the gene that, when silenced, stopped the whole process, was for a protein called ZP3. Zonapellucida glycoprotein 3.
Wait a minute, ZP3. Isn't that the sperm receptor on an egg?
Yes, that's the one.
You're telling me that a dying cancer cell is using a protein famous for its role in fertilization to trick the immune system.
It is an absolutely incredible example of evolutionary hijacking. A reproductive protein being co-opted for immune suppression. It's a total shock.
That's wild. Okay, so what was the evidence that this molecular handshake was really happening?
Well, first they confirmed the physical binding with techniques like immunoprecipitation. They could literally pull the GPX4-ZP3 complex out of the mix.
So they could see them stuck together.
Yep. Then they used computational modeling to predict the exact amino acids on each protein that we're touching. And to prove it, they mutated those specific contact points.
And the binding stopped.
The binding was destroyed, and so was the immunosuppressive function. No more IL-10. It was definitive proof of the structural basis for the sabotage.
Okay, so GPX4 binds to the sperm receptor ZP3 on a dendritic cell. But what does that connection actually do inside the cell? How does it flip that off switch?
It triggers an internal signaling cascade, specifically the cAMP-PRKA pathway. You can think of this pathway as the master power switch for the DC's engine room.
And the GPX4-ZP3 binding just flips that switch to off.
It does. And the engine for an activated DC, what it needs to mature and do its job, is a process called glycolysis. It's how it rapidly burns sugar for energy.
Right. Without that burst of energy, the DC can't mature. It can't travel to the lymph nodes to present a threat to the T cells. It's dead in the water.
Precisely. And this PRKA activation directly inhibits a key metabolic enzyme called PFKFB2.
So that's the main gas pedal for glycolysis.
It's the gas pedal, and this pathway puts a boot on it. They confirmed this by measuring that the DCs were consuming less sugar and producing less lactate, classic signs of suppressed glycolysis.
So the end result is the DC is metabolically crippled, it can't mature, it can't migrate.
And because of that, T-cell priming is defective. The whole anti-tumor response grinds to a halt before it even begins.
And this isn't just happening to any DC, right? It's specific.
That's a critical point. The suppression seems to be primarily restricted to a subset called CDC1s, and those just happen to be the immune system's absolute best cells at cross-presenting antigens and activating killer T cells.
So the tumor is specifically targeting these special forces of the immune system.
By starving them of their energy source.
So let's recap this part. A dying tumor cell releases GPX4. It finds the ZP3 receptor on the most important immune cell, the CDC1.
Right. That binding hijacks the cell's internal signaling to shut down its metabolism.
Which prevents the DC from maturing and activating the T cells that are supposed to kill the tumor. It's an incredibly elegant portrayal.
It really is a complete metabolic shutdown.
This whole mechanism, as sinister as it is, really presents a massive therapeutic opportunity.
A huge one.
Because if we can just block this new metabolic immune break, we could potentially boost the effectiveness of a whole range of existing cancer therapies that we know induce ferroptosis.
That's the payoff, absolutely. And the researchers went on to test exactly that in their preclinical models by blocking either ZP3 or the PRKA pathway.
And the results were pretty compelling. Let's start with standard chemotherapy.
Okay, so take pancreatic cancer, PDAC. The standard chemo regimen is gemcitabine plus nab-paclitaxel, or GP. They found this regimen does, in fact, cause GPX4 release.
So it's creating its own resistance mechanism.
To an extent, yes. But when they added an anti-ZP3 antibody to the mix,
The survival in the mice improved.
significantly improved. And crucially, when they depleted the mice of their CD8 plus T cells, that survival benefit disappeared.
So it proves the blockade wasn't just killing more cells directly. It was working by unleashing the T cell.
It was re-engaging the immune system. And they saw similar things with cisplatin in models of ovarian, lung, and bladder cancer. It seems to be a general principle.
So it works with chemo, but what about with modern immunotherapy? That's the big question.
Right. So they looked at the frontline treatment for non-small cell lung cancer, which is a combination of pembrolizumab, that's an anti-PD-1 drug plus chemotherapy, PCP.
So you're talking about making the cornerstone of modern cancer treatment, anti-PD-1, work even better.
That's the goal. And they found that, yes, this PCP chemo combo causes GPX4 release. And in vivo, when they added the ZP3 antibody,
It enhanced the efficacy of the whole regimen.
it did. And again, it was entirely dependent on the CD8 plus T cells. This tells us that the GPX4 ZP3 axis is an active resistance mechanism that is likely limiting how well our best immunotherapies work right now.
This is all really powerful preclinical data, but what about in humans? Is there any clinical evidence that this is actually relevant to patients?
There is, and it's quite strong. They looked at blood samples from patients with pancreatic cancer being treated with that GP chemotherapy.
Okay.
The patients who didn't respond to the therapy had significantly higher levels of circulating GPX4 in their blood compared to the patients who did respond.
Wow. So the break is literally measurable in the blood of non-responders.
Exactly. And even more, when they looked at tumor biopsies, those same non-responders had much more ZP3 co-localizing with dendritic cells right there in the tumor.
The spy and the target are caught together at the scene of the crime.
It's a perfect way to put it. And there's more.
What about ZP3 as a biomarker on its own?
They analyzed the Cancer Genome Atlas data, a huge public database, and they found that high ZP3 expression correlates with poorer overall survival in 9 different types of solid tumors.
Including pancreatic and lung cancer.
Yeah, including those, yes. And in those tumors, high ZP3 also correlated with fewer CD8 plus T cells getting into the tumor. The more ZP3 you have, the worse your immune system is at fighting back.
That's an incredibly strong clinical link. It really moves ferroptosis from just being a type of cell death to being,
Right.
well, an active immune modulator.
It is. This GPX4 ZP3 axis is, for all intents and purposes, a new type of immune checkpoint, but one that works by controlling metabolism.
And if you can block it, you restore the DC's ability to burn sugar, to mature, and to kickstart that whole T cell response.
You turn an immunosuppressive event into a powerfully immunogenic one. It's targeting that metabolic immune interface.
Which brings us to our final provocative thought for you to take away from all this. The GPX4 ZP3 mechanism works by specifically targeting the energy supply of the dendritic cells shutting down glycolysis. We know the entire field is racing to find the next big checkpoint inhibitor. So the question is this: how many other metabolic immune checkpoints are tumors using right now to evade our best treatments? And how can targeting a cell's energy supply directly become the next pillar of cancer immunotherapy?
It may be that the future of cancer treatment isn't just about what kills a tumor, but about what powers the immune cells that do the cleanup. We'll keep diving into that intersection.
Thank you for joining us for the deep dive.